Hello, my name is Rachel Denison, and I'm really happy to be here today at AMP2020. Thank you so much to the organizers for the invitation and for organizing this great meeting. I'm really excited about it. And I'm excited to tell you today about our work modeling pupil responses to dynamic events. So this is a view of someone biking through Midtown Manhattan. And this helps us appreciate that our environment is highly dynamic. We must perceive and act in real time based on a constantly changing stream of visual input. And today, I'd like to ask how we might use the pupil as a window into this kind of dynamic processing. So pupil size is an accessible online indicator of perceptual and cognitive processes, which has potential to reveal continuous processing dynamics, including anticipatory dynamics that are not accessible from behavior alone. So in my talk today, I'm going to be focusing on these perceptual and cognitively driven pupil responses that occur even in the absence of luminance changes. However, whereas the neural dynamics that drive the pupil are fast, pupil dynamics are relatively slow. So it takes about 2 seconds for the pupil to dilate and constrict in response to a stimulus. And this poses a challenge for linking changes in pupil size to specific events in rapid sequences. One strategy for addressing this challenge is to model the relation between perceptual and cognitive events and pupil size. And such models linking pupil size to trial events have been used in the literature, but not yet systematically evaluated. And as a result, there is currently no standard approach to pupil modeling in the field. So our goal was to develop and evaluate models linking pupil responses to rapid sequential events. So first, let me introduce the data set that we used for the model development. We used data from one of our previous papers on temporal attention, which is the prioritization of sensory information at specific points in time. In the temporal attention task, participants were fixating on the center of a computer screen, and they were asked to judge the orientation of grading stimuli that appeared here in the lower right quadrant. The whole time we were tracking your eyes using an iLink 1000. So in the temporal attention task, we have a trial sequence that looks like this. 2 targets were presented sequentially in the same location. These are T1 and T2. And these could, had independent orientations, and they were separated by a stimulus onset asynchrony, or SOA, of 250 milliseconds. Before the targets appeared, there was a pre-cue, which was a tone, which instructed observers to attend to T1, T2, or both. And after the targets appeared, there was a response cue, which instructed them to report either T1 or T2. And on 75% of trials, the response cue matches the pre-cue, so observers had an incentive to attend to the target that they were later asked to report. For the purposes of this talk, it's not critical to remember too much about this temporal attention manipulation. The key thing here is to note this trial sequence, which is multiple sequential events within a short trial that was itself less than 2 seconds. It is important to note that observers also had a cognitive task that they had to perform, either a discrimination task where they reported the orientation of the target as being either clockwise or counterclockwise, or they, in other experiment, observers performed an estimation task where the orientation of the target could vary randomly, and the observers were asked to report the exact orientation by adjusting a probe stimulus. So again, this task was useful for our pupil modeling goals because it has multiple trial events in a rapid sequence with no changes in overall luminance, along with two cognitive manipulations, a temporal attention manipulation and a task manipulation, where an estimation task would be expected to have a higher cognitive demand than a discrimination task. So first, here are the raw data. So here are the pupil time series for individual observers performing this task. Here I am plotting the pupil area as a percent change from baseline across time. The two tasks correspond to either the filled or open lines, and different colors in this plot correspond to different observers. Here we've plotted the three temporal attention pre-Q conditions separately for each observer, so you can get a sense of the consistency of these pupil time series within observers across three conditions. So for example, here is the three conditions from one observer. And here we had 21 data sets, 9 for discrimination, 12 for estimation. 3 observers completed both tasks. And what I want you to notice in this plot is that the pupil time series varies a lot across observers, but it is very reliable for a single observer in different conditions. So we used a general linear modeling framework to model the pupil time series across a trial. And we did this on an individual observer basis, which is motivated by the fact that we do see this very reliable pupil time series within individual observers. So let me explain this general linear model framework. Here I'm showing again the trial timeline, time on the x-axis. The physical stimulus events are in gray, so that's the pre-Q, which was an auditory tone, T1 and T2, which were gratings, visual gratings, and the response Q, which was another tone, auditory tone. To construct the model, we imagine that each stimulus event drives the pupil to dilate. And we model each of these events with a delta function with some unknown amplitude, which reflects how much the event drives pupil dilation. And we also model the latency of these events, which reflects the timing of the neural response that drives the pupil response to that event. So for example, this purple event here has a slight positive latency, which would express the idea that the neural event driving the pupil that is associated with this response cue happens around the same time as the response cue itself. Here we have a delta function that is occurring in anticipation, the corresponding event T1. So this would, the idea here is that the event, the neural event driving the pupil could actually occur before T1, and that's possible in this task because the timing of trial events is predictable. So we allowed in this case for both positive as well as negative latencies. And again, negative latencies mean that the neural event driving the pupil could anticipate the stimulus event. So we call these stimulus-associated delta functions, the stimulus-related components of the GLM, and again, these are shown in these colored lines here. So next, some previous authors have included, not only stimulus-related components, but also a task-related component, which is sometimes called a decision-related component, shown in black here. So here we imagine that some cognitive process starts at the beginning of the trial and then is sustained until the observer makes a response. So the timing of this sustained task-related component is fixed, but the amplitude can vary. And so this part of the model, here that I'm showing here, reflects all the perceptual as well as cognitive events that we hypothesize are driving the pupil to dilate. So importantly now, the pupil does not respond instantaneously to a given event. Instead, it takes some time for the pupil to dilate and return to baseline. This impulse response function was measured by Hooks and Levelt in 1993, and it is well described by a gamma function. We are going to call this the pupil response function, or PRF. Hooks and Levelt found that while the form of the PRF is similar across observers, the exact timing can vary. Some observers' pupils dilate more quickly and some more slowly. So here we let the timing of the PRF peak vary with this parameter Tmax, which reflects the time to peak. Okay, so using these two ingredients that I just described, a model of the trial components and a model of the pupil response function, we can predict how the pupil will respond to a trial sequence by convolving the PRF with these trial components and then summing the components together. So in this plot on the right, I'm showing the convolved components in these colored lines. So these colored lines correspond to the trial events in the model of the trial components. And then we sum these together, and that produces for us this gray dotted line. In yellow here is the actual data this particular model was fit to, so that's this yellow line, kind of closely following the model prediction. And so you can see here that the model provides a very close match to the data. Okay, so what I just showed you is the general framework for GLM modeling of the pupil response. But as you can see, there are different choices about what components and parameters to include in this model. And if we're trying to develop a standard model that can be used widely in the field, we must then ask, what components should we include in the standard model? Here we tested a few possibilities. So we tested timing parameters, including event latency. So should we allow the modeled neural events to occur at different times than their associated stimulus events, or should they be completely locked to their stimulus events? And the PRF time to peak. This parameter also I might refer to as Tmax. So should we use the standard number from Hooks and Levelt, which is 930 milliseconds? That was based on a population average, and that's a parameter that many, possibly all pupil modeling studies today have used. Or should we let this number vary? Task-related component. Should this task-related component perhaps associate with a cognitive process underlying decision-making be modeled as a boxcar, as I had shown in the previous plot, that kind of square boxcar function? Should it be linear, kind of ramping up slowly over time? Previous modeling work has used both of these. Another question is, do we even need this component? Is it sufficient to model the pupil responses to the trial events, or do we gain something by including this kind of sustained task-related component? So to answer these questions, we used model selection. We compared 24 models that included different combinations of parameters in a full factorial model comparison. We tested for the two timing parameters, latency and Tmax, the task-related component here, and a fourth parameter, the y-intercept, which is just a baseline parameter. So the table, this table at the bottom here, shows all the different models, and it shows which parameters were included in each model. So the columns here are models corresponding to these numbers above here. And the pluses, these little pluses, indicate that the parameter was included in the model. And then here for the task-related component, B corresponds to a boxcar, and L corresponds to a linear task-related component. And then a blank space means that the parameter was left out of the model or set to a fixed value. So we then fit all of these 24 models to our data and measured the goodness of fit using BIC scores. And that's what we're plotting here. So we're plotting those BIC scores here as the difference from the best model, with worse fitting models having higher delta BICs. Colored points here are individual observers. Black markers show the group mean and SEM. And the best model, model number 1, shown here, included all of these parameters, included the latency, the y-intercept, the Tmax, and the task parameter, in fact, the boxcar, which indicated that these timing parameters and task-related component do improve the model fit. So these improve the model fits and suggest that these should be included in a kind of standard model. Okay, the next question is whether fitting the model actually produces meaningful parameter estimates. So this is important because if you're doing an experiment, you often want to interpret these parameters. For example, you might want to ask whether the amplitude of the pupil response to a specific stimulus differs across experimental conditions. And if the parameters can't be estimated correctly because of, for example, some redundancies in the model or some issue with the fitting procedure, then your parameter estimates would be uninterpretable. So to address the question of the interpretability of the parameter estimates, we did a few things. First, we performed parameter recovery, and that's what I'm illustrating that process here. So we selected various parameter sets at random, so we know the ground truth of the parameter values. And then we used the best model to simulate pupil time series data from these known parameter values. We then fit the model to these simulated time series and asked whether the fitted model parameters matched the original parameters that we started out with, whether they matched the ground truth. So here are the results of that parameter recovery analysis. Each subplot here is a 2D histogram where the x-axis is the actual parameter value that was used to generate fake data, and the y-axis is the parameter value that was estimated by the model fitting procedure. So a solid diagonal with a correlation value of 1 would indicate perfect parameter recovery here. And here I'm plotting each subplot corresponds to each parameter in this model. So parameter recovery was overall quite good, but there were some important differences among parameters. So the timing parameters tended to have better model recovery. So that includes these 4 latency parameters associated with each event, pre-queue, T1, T2, and response queue, as well as the Tmax parameter, or this time to peak of the pupil response function. So these timing parameters tended to have better recovery than the amplitude parameters, with almost perfect recovery for Tmax, this time to peak parameter. Of the amplitude parameters, recovery for T1 and T2 was lowest, likely because these events are quite close together in time. They're 250 milliseconds apart, which may introduce some ambiguity during fitting. So a parameter recovery analysis, which you can do even before you run your experiment, gives insight into how the trial timing will affect the reliability of the parameter estimates. And this analysis here suggests that even for a rapid trial sequence, the fitting procedure is producing fairly reliable estimates. The second way we evaluated the reliability of the parameter estimates was to ask how uncertain the estimates were in the data itself. So to assess this, we bootstrapped each observer's data 100 times and refit the model, which allowed us to calculate confidence intervals for each parameter estimate for each observer. So here I'm showing you the parameter estimates for an example observer. So again, we have the amplitude parameters for each trial event, this task or decision-related parameter. Here we have the latency parameters for each trial event, and then again, Tmax. And here the box and whisker plots here are showing the 50% in the box and 95% in the whisker confidence intervals for each parameter. And if we look across our group of observers, the mean uncertainty as measured by the 95% confidence interval was about 2.5% for amplitudes and 350 milliseconds for latencies. Finally, we evaluated the reliability of parameter estimates by examining their consistency across conditions, that is, across independent sets of trials for each observer. So these plots here show the different parameter types. Again, amplitude on the top, latency, and Tmax. Again, colors correspond to observers, and individual dots here correspond to one condition for one observer. So if dots of the same color are close to each other, that indicates that a parameter estimate that is consistent across conditions. And what we found, as you can see here, is that parameter estimates were much more consistent within observers than across observers. And in fact, there was considerable variability across observers. So this pattern was very clear, I think, for Tmax, where we see this value, this estimated Tmax value, very consistent across independent sets of trials for single observers, but varying over about 800 milliseconds across observers. And this really drives home the point that different observers have different pupil dynamics that need to be taken into account to interpret the pupil time series. Finally, then, we asked whether any of the experimental manipulations that we did affected either the amplitude or latency of the pupil response as estimated by the model. And we found that all the event amplitudes were higher in the discrimination task than in the estimation task. So what I'm plotting here is the estimated event amplitude for these different trial events, pre-Q, T1, T2, and response Q, in these two different tasks. So the filled circles are discrimination, the open circles are estimation. So all of these event amplitudes were different for the two tasks. The task-related amplitude did not differ, the y-intercept did not differ, and the latencies did not differ. So this is a finding that's specific to these amplitudes. We think it's possible that we found higher discrimination amplitudes compared to estimation amplitudes because of the higher cognitive demand in the estimation task. This could be because the pupil is more dilated at baseline in the estimation task because of this higher cognitive demand. So it actually increases less during the trial itself. This scientific finding is potentially interesting. It needs to be followed up on, but for our methodological purposes here, this finding demonstrates that the modeling approach is sensitive to cognitive manipulations of this kind. Okay, so to conclude, how are we doing in terms of our goal of developing a standard pupil modeling framework? So what we found here is that, first of all, pupil responses to rapid trial events can be distinguished using GLM modeling, even though the pupil impulse response function is much slower. Individual observers, have consistent but idiosyncratic pupil dynamics, which should be modeled at an individual observer level. We found that timing parameters, including event latency and time to peak, improve the model fit, and so does a sustained task component. So this suggests that a particular model structure should be used to analyze pupil data. And finally, we found that cognitive manipulations can affect specific pupil parameters. So our conclusion here is that pupil modeling with GLMs links pupil responses to underlying sensory and cognitive events. This can be done even for rapid event sequences that are themselves faster than the sluggish pupil response. And importantly, doing this kind of modeling can affect the interpretation of your data. So, for example, what looks like an amplitude difference in the time series could be not because the pupil responded more to the previous event, but because, for example, the reaction time is longer. So the sustained task component would tend to increase the amplitude of the pupil response. Modeling can distinguish these two possibilities, again, allowing us to link pupil responses to their underlying sensory and cognitive events. So, especially as this workshop is aimed at methods development, there are a few limitations of the present work as well as future directions that I'd like to finish up by discussing. So first of all, these models all general linear models assume that the pupil responds like a linear system. This assumption is based on supporting data from Hooks and Levelt, but more work should be done to test this assumption in a range of conditions and determine if and when this assumption breaks down. For example, if the pupil is already quite dilated, it may have a limited ability to dilate further. And this type of situation would create nonlinear pupil behavior, which would be important to account for in modeling. Second, we realized that when we were working with these models that, at least to our knowledge, it's unclear what kind of noise pupil data is subject to. And a better understanding of the noise properties of the pupil system would improve our ability to simulate and model pupil data and to derive useful metrics from procedures like parameter recovery. Third, we tested these models on a data set that was well suited to our goals and that it had multiple trial events in a rapid trial sequence. But it will be important to compare these models in other data sets and other tasks to see if the main conclusions generalize and to assess the reliability of parameter estimates with other trial structures, other trial timings. I think there are also really interesting opportunities to compare parameter estimates across tasks for the same individual observers. For example, if time to peak were estimated using a very simple task with just one stimulus presented, could that estimate then be used to predict pupil time series from trials with rapid event sequences? So to facilitate pupil modeling for a wide variety of tasks and data sets, it was important for us to make our modeling and model evaluation code widely available. So to that end, we have released a software package called PRET, which stands for Pupil Response Estimation Toolbox. Here's the GitHub address at the top of the slide. On the right here, you can see a movie of PRET model fitting in action, which is a visualization that is part of the toolbox. So PRET is an open source MATLAB toolbox for GLM pupil modeling. And it performs all the analyses that I described today, including basic preprocessing and model specification, model fitting, bootstrapping of parameter estimates, data simulation and parameter recovery, and model comparison. So it is intended to be a complete package for model fitting as well as model comparison and uncertainty estimation. This package is really aimed and intended to facilitate continued development of some field standard pupil modeling approach. And we've tried to make PRET accessible and user-friendly. So, even if you haven't worked with these modeling techniques before, you should be able to try them out on your own data. We hope pretty easily. So with that, I'd like to thank the people who contributed to this work. So I performed these experiments and did this work while I was a postdoc at New York University working with Marisa Carrasco. Jacob Parker was an undergrad RA in the lab who worked with me on this project and was the lead developer of the PRET toolbox, this MATLAB toolbox, and it's really nice. So if you use it and like it, that's all thanks to Jake. And now I've moved to Boston University, where I'm starting my own lab. So thank you so much, and look forward to discussing more. Thanks.